Welcome to NOAA Live Alaska. My name is Lisa Ruki-Raring, and I'm going to be moderating today's webinar. This series is a collaborative effort by NOAA Fisheries Alaska Fisheries Science Center, where I work, NOAA's Alaska Regional Collaboration Network, and NOAA's National Weather Service. Today, we're joining with our sister webinar series, NOAA Live, which is sponsored by NOAA's Regional Collaboration Network and by Woods Hole Sea Grant, which is located at the Woods Hole Oceanographic Institution on Cape Cod, Massachusetts. Both the NOAA Live and the NOAA Live Alaska webinars are designed to help you get to know NOAA and some of our incredible experts. All of our speakers work for some part of NOAA, the National Oceanic and Atmospheric Administration, which studies the ocean and the atmosphere and where the two meet, from weather to ocean to the animals that live around us. During the month of March, the NOAA Heritage Program is helping us take you behind the scenes at different NOAA facilities during our NOAA Open House Series. If you've missed seeing these tours live, the recordings are available on the NOAA Live website. We have 2 tours left in March to travel virtually across the country to showcase some of the amazing places where our scientists, engineers, educators, technicians, and interns work. Today, we're visiting NOAA's Alaska Fisheries Science Center's Kodiak Laboratory in Kodiak, Alaska. While we'll be talking about NOAA's role in research and stewardship, we want to recognize that we're all coming to you from the traditional lands of Native communities who have substantial Indigenous knowledge and much to share with us. We respectfully acknowledge that our speakers are coming to us from Kodiak Island, the Alutiiq-Sugpiaq homeland. We thank and acknowledge the tribes of the Kodiak-Alutiiq region. The heritage and culture of the Alutiiq people continue to enrich the community. We'd also like to acknowledge that we're hosting this webinar from the traditional lands of the First People of Seattle, the Duwamish people past and present. We also want to recognize and thank our American Sign Language translators, Crystal Butler and Carol Tipton, who will be translating for us today. A few guidelines before I hand you over to our speaker. You're all muted because we have a lot of people on the line and we want to make sure that everyone can hear our speakers. However, there's a box where you can write questions. Please let us know if you're a class tuning in, and if there are multiple people watching, let us know who's asking the question. We encourage you to ask questions as we go, and my colleague Chris Beyer and I will be keeping track behind the scenes for our speakers. We may not get to all of our questions, but we'll try to answer as many as we can. All right, I will hand it over to our speakers today, Ali Conrad, Chris Long, and Switgard Dustelov, to introduce themselves.
Thank you so much, Lisa. Yeah, hi everybody and welcome. I'm Ali Conrad. I'm a fisheries biologist, and I'm also the Seawater Lab coordinator here at the Kodiak Lab. And I've got a couple of my really good coworkers with me here today, and they'll introduce themselves next. I'm going to start off by telling you how I became interested in ocean science. So I was actually born and raised here in Kodiak, and I grew up right outside the ocean. Like, the ocean was right outside my front door. So I became interested in the ocean at a really early age, and I think that sparked my whole interest in this career field. Svitgard, how about you jump on and tell us what you think and how you became interested in ocean science?
Hi, I'm Svitgard Dustelov, and I am a contractor for NOAA, working with the percolator, as I will talk about later. And I became interested in ocean sciences as a kid, watching movies with Jacques Cousteau going diving on TV. I grew up in Germany, so I grew up far away from the ocean, and now many years later I find myself here in Alaska in Kodiak, and I'm just really excited to be here today. Right.
Ali, we're getting a little bit of echoing, so I was wondering whether you might be able to mute your microphone.
Hi, my name is Chris Long. I'm a research ecologist here at the Kodiak Lab in NOAA, and I actually grew up in the Kalahari Desert, of all places. And so this, you could probably interpret my switch over to marine biology as a rebellion against my parents. But actually, as a child, I loved looking under rocks in the desert, and we would find scorpions, and we'd find lizards and snakes and all sorts of cool things. And here in Kodiak, I look under rocks and I find other cool things, you know, crabs and sea slugs and all sorts of nifty things here. So it's really not all that different, just a lot more wet.
That's great. Thanks, Chris. So this is where we're talking about today. This is Kodiak, and this is the facility. So we work in the Kodiak Fisheries Research Center. Locally, this building is just called the Touch Tank, since we have a public touch tank and aquarium. This facility is actually owned by the Kodiak Island Borough, and it was built in 1998. Part of the funds that were used to complete the build came from the Exxon Valdez oil spill settlement. NOAA has a lease for the space that we occupy in this building, and we're accompanied by some really, truly incredible other organizations as well, including the Alaska Department of Fish and Game, the International Pacific Halibut Commission, Kodiak Regional Aquaculture Association, and Kodiak Soil and Water Conservation District, as well as the Ocean Science Discovery Program, which we're going to talk about in a little bit. In a non-COVID year, we have about 10,000 visitors that come through our front doors. So a big part of our time is spent communicating with the public and doing outreach, and it's something that we truly love. So next, we're going to see a video of walking into our building. Bear with us if it's a little slow this morning. Our internet here in Kodiak is a little bit slow today. So this is the outside of our facility. We're just gonna walk right up to the front doors so you guys can get a feel as if we were walking right into the building for our lab tour. Like a lot of other places, our building is closed to the public right now, so the only ones of us that are in here are people who are here to do mission critical science activities. We have a lot more posters on our windows than we typically do, but this is our absolutely beautiful lobby area with our whale skeleton and all our different posters and our big map of Kodiak. So one of the coolest parts about our facility, we have lots of different spaces and types of rooms, but definitely one of the coolest parts is our Seawater Lab. There's actually 2 Seawater Labs in the building. One is occupied by NOAA and one is occupied by the Alaska Department of Fish and Game. So I'm going to play this video as well. It might be a little jerky as the last one was. But if you were to head downstairs from my office right now, you would walk right into the garage. Our garage looks like a pretty standard garage. It's full of all sorts of tools and then also fisheries equipment.
Allie, I think we're getting a little bit of an echo. I wonder whether Chris might be able to mute himself.
Okay.
Okay, so now you're okay. Now you're muted, Allie.
Okay, let's try that again. Does it sound like there's an echo so far? Okay, well, we'll keep going and we'll see how it sounds. So like I said, this is the garage. We've got some pieces and parts of our aquarium in here, which we'll talk about in a little bit. And then you step into the wet lab, and here's one of our fisheries biologists, Connor Cleary. He's setting up a new experiment in the lab. We're also going to take a look into some of our large tanks. So this first one we're going to look in is a fish tank. These are fish that we have for our aquarium. There's different types of rockfish, copper rockfish, black rockfish. This is a greenling right here, another greenling. And then let's peek into another one. This is also a fish tank. We've got some flatfish. This is a halibut back here in the back. There's a yellowfin sole, a rock sole. This is another copper rockfish. And then back here in the back, we've got a kind of just a pile of sculpins hanging out on some rocks. Now we're going to take a look inside a crab tank. So these are red king crab that came from Norton Sound. Norton Sound is the part of the ocean that's where Nome, Alaska meets the sea. And there's both male and female red king crab in this tank. Look at the size of these crab here. We're going to see some more crab in a second, and they are different sizes. So this tank, tank 16, as we walk up to it, these are also red king crab, but these are Bristol Bay red king crab. And you'll probably notice here, these crab are fairly large. They're quite a bit larger than the others, and that was just purely because of the experiments that they were in. So they were selected for a specific size for their experiments. Excuse me. This really neat piece of equipment is called the crab condos, and it's a tank system that holds quite a few little individual tanks. We're going to zoom in on the tank L4 here, and right there in the middle, that's actually a baby red king crab. That king crab is about 1 year old. This last part of the lab that we're going to look at is just kind of an extra area full of tanks where we keep extra animals for the touch tank in the aquarium. So these are actually fairly bare right now. Typically these tanks are quite a bit fuller, but we've been operating such a limited capacity that we haven't been doing many collections, but you'll see lots of sea stars, sea cucumbers, anemones. There's some snails in here, a hair crab, leather stars, some more sea stars. There's also a very large urchin on the right there, some tube worms, excuse me. And then this is a cool tank. It is just full of a bunch of different sea stars and a very large urchin. And I just love this video to see all the different colors and textures all in one place. It really shows the diversity of animals. So if you walk out of the wet lab, the seawater lab, and you walk into the dry lab, it looks quite a bit different. This area is full of specialty equipment and instruments. And this is where we process samples. So this is actually Joletta Silva. She's one of the biological technicians here for the lab. And she was processing samples and putting them in that small drying oven. Once we walk over into this other part of the lab, this is Svitgard's area, and this is the percolator. And she's going to talk a lot about this piece of equipment here in a little bit. So if you were to walk out of the dry lab downstairs, you would walk into definitely another one of the coolest parts of this building, and that's where the aquarium and touch tank are. So the NOAA staff is super happy to be able to work cooperatively with the Kodiak Island Borough in maintaining the aquarium and touch tank. The borough actually maintains the tanks themselves and the infrastructure, and the NOAA biologists, myself and Chris and Svitgard and a lot of other people here, we take care of the daily husbandry tasks like feeding and cleaning. NOAA also holds the permit so that we are allowed to collect and hold those animals. Unfortunately, the building's closed right now, like I said. But if you were to walk in today and look at the touch tank and aquarium, it would actually look like this. We're remodeling. So the tanks are empty and dry. In this photo, you can see Eddie. He's the building maintenance engineer for the borough, and he's pressure washing the structure inside the aquarium. We've done a bunch of deep cleaning and buffing the glass, and we've actually made some upgrades to the structures inside the large aquarium. It's been a lot of work, but it's really fun, and we're so excited to share our bright, shiny new aquarium with you when we're back open to the public. So lastly, another really great program that happens in our building is the Ocean Science Discovery Program. Svitgard is actually the director of this program, and she does that in addition to her job as a scientist with our lab. So each spring inside the Ocean Science Discovery Lab, Kodiak school-age kids come to our facility for a one-day program with their class to learn about our oceans and the animals that inhabit them. The NOAA biologists at our lab love helping with this program. It's really fun, and we really enjoy our interactions with the local school kids. So now that I've showed you all around our lab and I've told you a bit about what we do, I have a question for you guys. How many individual live animals do you think we have in the Kodiak lab right now? It's one of these 4 options. So send your guess in the chat box and Lisa will read them off.
All right, so I see that some people are already putting in their guesses as to how many animals there are. Texas is saying D. Grace is saying D. Hunter is saying C. Charter is saying D. So is Theodore. We've got a lot of C's and D's. And then Jillian thinks 17, so that's A. But it looks like most people are saying either C or D.
Good job, you guys. Yeah, so a lot of you were right. So C is actually the amount of animals. 526 animals is what we have live in the lab today. The answer D I actually put on there because it's my best guess for about how many animals we'll have in about 2 weeks from now. So there's a boat out in the Bering Sea just today, and they're collecting crab for us for upcoming research projects. So here in another couple of weeks, we will just about double the amount of crab that we have in the lab. So that's all that I have for you guys about general tidbits about our lab. Before we jump into the next section, I've got just a little bit of time. I can answer a few questions, and then we'll throw it over to Chris, and he'll talk about his section.
Okay, well, we do have some questions about the lab tour that you just showed us. Janet was wondering, why do the large tanks have covers on the top?
Yeah, that's a good question. We put covers on the top for a few reasons. Sometimes the project is light dependent, and so we want to make sure that we can control the amount of light that goes into the tank. We also put covers on because crab are bottom dwelling animals and they're used to it being relatively dark.
Great, and then Michelle was wondering, why are there 2 seawater labs?
Yeah, that's a good question too. So that's just how this facility is set up, is that there's 2 seawater labs. So I showed you a walkthrough of ours, but then ADF&G also has their own. Their lab is probably about half the size of ours. It's quite a bit smaller of a footprint, and they run their own crab projects in there. It's really cool to have them just right across the hall.
And for those of you who are from out of state, ADF&G stands for Alaska Department of Fish and Game. So they're the state organization that studies fish and game around the state of Alaska. Sarah was wondering, how big can king crabs get?
Oh boy, how big do king crabs get? I don't know off the top of my head. Yeah, Chris is doing it with his arms. Chris, what do you think? How big can they get?
So on the survey, carapace length, so measuring from the eye socket to the back of the carapace, will get above 200 millimeters. What I was doing there was kind of the size of some of the biggest king crab, kind of from leg tip to leg tip that you would get. So, you know, maybe five feet across there.
Wow, that's really big. So Mr. Westbrook's class was wondering whether the Kodiak Lab has any volunteering opportunities.
Yeah, we do. You know, when we're open as business as usual, we do have some volunteering opportunities. So I'm not sure exactly of the avenue that you would have to go through, but probably getting a hold of Lisa, and she would get you in the right direction to who you need to talk to about volunteering opportunities.
Great. And then Kaylee was wondering whether the animals ever scare you in the saltwater lab.
Yes, they do scare us sometimes. That is such a good question. The fish, mostly, I would say, are pretty can scare you. Like every now and then you'll open the lid up and one will jump and it'll kind of take you by surprise. The only other scary thing I think is when you get your finger pinched. That's kind of scary for a few moments there.
All right. And then Charter was wondering, what is the most amount of crabs you've had in 1 year?
Oh, wow, that's a good question. I don't know what our max capacity is for the lab. What do you think, Chris?
We've had upwards of 10,000 juveniles in the lab at one time when we've reared them. And if you count larval crab, you're going probably tens of thousands.
Yeah, that's a
larval
very
crab or 2 millimeters.
Yeah, we have run experiments on larval crab, and the king crab, once they hatch out hundreds of thousands of larvae, and we've had all those larvae in the lab at once. So a lot if we really go down to the very, very teeny tiniest crab.
And we've got a bunch of questions about what kind of research you do. So maybe that would be a good segue into talking about our next section.
Yeah, that's a super good point. Let's throw it over to Chris and he can tell us about his research.
Thank you, Allie. So I've been working in Kodiak for more than 11 years at this point. One of the big topics that we've been studying here ever since I got here is looking at this process that's called ocean acidification. Now, ocean acidification is the gradual increase in the acidity of the oceans, which is caused by increasing carbon dioxide concentrations in the atmosphere. And this is illustrated really well in this graph I've got here. So this is some data that's been collected in Hawaii by some NOAA partners out there. And what the red line is showing you on there is the concentration of carbon dioxide in the atmosphere. And this graph is running from, you know, the mid-1950s all the way up to the 2010s here. And what you can see is that we've had this gradual increase in carbon dioxide, and this is due primarily to burning fossil fuels, so gas and coal and stuff like that. As the concentration in the atmosphere has gone up, that gas also dissolves into the ocean. So if you look at the green line on this graph, that shows the carbon dioxide concentration in the ocean by Hawaii as well. And you can see this time series, it starts a little bit later in the 1980s, but you can see that it follows the trend of the atmosphere really closely. And as the atmosphere has gone up, as you would expect, the concentration in the water has gone up as well. Now, the thing about carbon dioxide is it doesn't just stay dissolved. It actually reacts chemically with the water and produces an acid called carbonic acid. And so as you dissolve more and more CO2 in the water, the pH of the water goes down, or the acidity goes up. And that's what the blue line is showing you right here. So as the concentration of CO2 has increased, the pH has gone down. And over the last about 100 years, we've seen about a 0.1 reduction since the beginning of the Industrial Revolution. And that might not sound like a lot, but actually from an organism's perspective, even from your perspective, if your blood pH changed by that much, you'd be in the hospital. So that is a big deal. So in general, just the concentration of CO2 is going up in the atmosphere. It's going up in the oceans, and as that happens, the pH is going down. So next slide, please. So why do we care about this? Well, we care about this because there's a lot of animals in the ocean, and many of them have shells that are made out of calcium carbonate. So what I'm showing you pictures of here, these are snails that were held in experiments, and they were held at different pHs in the lab. And as you go kind of from the left hand of the screen, where you see these nice healthy snail shells, to the right hand side, that's what happens as you decrease the pH, as you increase the acidity. And the reason you start to see this is because calcium carbonate dissolves if you take the pH low enough. And so on the upper column here, you see, or upper row, you can see that the shells go from kind of nice and bright and shiny, and then as you go towards lower and lower pHs, you start to get cracks that are forming. You start to see areas of the shell that are worn away. In the lower row, you see actually holes starting to form. So again, you start with a nice clean shell, but as you get into higher and higher acidity, you start to have holes being formed in the shell. And of course, it doesn't take a rocket scientist to say that a snail with holes in its shell is probably not going to be very healthy. Next slide. So how does it affect some of our animals? So right now we're running an experiment, and this video is showing that experiment running down in the wet lab. I was actually down there collecting data on it on Sunday. So we've got 3 tanks down here, and what we're looking at is how does OA affect these little juvenile red king crab. So as you look at these tanks, each of those tanks has these little inserts in them, and in each of those inserts is a single crab. Now, each of those tanks now is set at a different pH. And this is a pH monitor that is keeping track of what the pH is in the tanks. And if the pH drops too goes too high, then there's a switch in there which bubbles pure CO2 into the tanks so that it drops the pH back down. Same process that is causing ocean acidification in the actual oceans here. So then every day we have to, of course, take care of our crabs. We hold them in individual containers so we can keep track of each crab individually and measure exactly how much it grows. So we check it every day, and when it molts, we're able to measure the crab and see how much it grew in between molts. Crabs are like, like almost all crustaceans, they have to molt to grow because they've got that hard outer shell. We also check every day to see if any of them have died. Because one of the effects we've seen is that you get an increased mortality rate. That means the crabs die faster if you put them in low pH or high CO2 water. So this experiment will be running for about a year in the lab. We've run it for about half a year so far. The other thing that Ali's showing you right here is as we measure the pH with that meter that was there, and we record it on a tablet, so that we know exactly what conditions the crabs were in on a regular basis. So as we look at how ocean acidification affects crabs, I already gave you kind of a hint of this, but this is from an earlier experiment where we were looking at two different species, red king crab, which is the small little guy in the upper right, and the tanner crab, which is the one in the lower left there. What we've got graphed here on these two plots, in the upper one, the scale on the y-axis runs from 100% survival, which is how many crabs were, with the percentage that was alive at the beginning of the experiment, all the way down to 0. And each of the plots has 3 lines on it. The solid line is the control, so that's just the pH that's out in the Gulf of Alaska right now. And then the other 2 are reduced pHs there. And what you can see is that the control crabs do pretty well. You get some mortality over time, but that's normal for small crabs. But as you decrease the pH, that mortality rate goes up. And so by the time you get to 7.8, now you can see that there's about half as many crabs alive by the end of the experiment as in the control. And in 7.5, they all die within 100 days. So very not good for little crabs there. Next slide, please. It also affects their skeletons. So crabs have an exoskeleton, which grows on the outside rather than our internal skeleton. And it also is made of calcium carbonate. So we did another experiment where we looked at larger crabs, and at the end of the experiment, we cut a small piece out of their carapace, which is the top of the crab right there. And on the left-hand side, you can see 2 images, one from crab that was taken from ambient pH, so that's good water, and then one from reduced pH. And this is the inside of the shell. So with the ambient pH, it's nice and shiny. You can see that looks nice and hard. There's no pitting. That looks like what you would expect from a healthy crab. But after spending time in reduced pH, you can see that now we're starting to get pitting. Those dark areas are areas where the calcium carbonate has actually dissolved, and so there's none left. Basically what's happening is the crab's shell is dissolving from inside out. This is kind of like the crab getting osteoporosis here. Similarly, not only does it give them osteoporosis equivalent of, it also makes them need dentures. So this is what Ali's pointing at right now is the claw of the crab. And this is what they use for grabbing onto things, for protecting themselves, but also for crushing the food they eat, such as clamshells and stuff like that. And if you zoom into the claw right there, you can see there are these little white bumps on the top. And those white bumps are called denticles. And if that reminds you of a dentist, it's because they both have the same root. These are basically little teeth, and they're really, really hard structures, which allows the crab to crush their prey. So the upper one, again, is the crab that is in normal, good water. And you can see those are nice, healthy looking teeth. They're white, they're hard, they're really bumpy. But after spending time in low pH water, those things have now dissolved away. And you can see that, basically this is a crab that needs dentures because it doesn't have its teeth anymore, they're gone. And so that crab obviously is gonna have a harder time getting food in the wild than the crab that has good claws. Next slide. So if I'm hoping we have time for questions, but I'd be happy to field any of these at least this time.
Yeah, I think we have some time for a few questions. So one of the questions that Jeffrey had was, what happens to the crab after the experiments? Do you let them go in the water?
So Alaska Department of Fish and Game regulates the use of animals in the lab. And one of the requirements that they have is that we're never allowed to let the animals go. And unfortunately for the crabs, for a lot of the things that we study, we have to kill the crab at the end of the experiment in order to measure them. So when we're looking at the exoskeleton, we have to sacrifice the crab before we're able to pull that shell off and look at it there. So that's pretty much we're not allowed to release the crab from the lab. Some of the crab, if we don't need to kill them at the end of the experiment, we'll use them in the touch tank and aquarium. So they go on and they can have a happy life of being fed and adored by the hordes of children we have coming in here.
And I'm sure that you have a lot of visitors during non-COVID times. Hannah wanted to know, you had mentioned osteoporosis, and she was wondering, what is osteoporosis?
So osteoporosis is in humans, and as people get old, often their bones become brittle, and it's because the cells that make the bones don't work as well as they used to when you were young, and so the bones lose density. That means there's not as much calcium carbonate in them, and they become easier to break. Yeah, it's just an analog. It's similar to what's happening to crabs in this.
Right. And actually, we had Oliver had a question about the snail that you showed, where the shell is eroding away, and he was wondering what happens to the snail after the shell erodes away.
I think a lot of the times they die. There's some evidence that they can kind of fix those shells if they're under those conditions for a short enough amount of time. But that shell is there to protect them, and so if it loses its integrity, then it makes them easier prey for other animals as well. Right.
So we had several people who had the same question. I think it was Stephen, Oceana, and Helena were wondering, how long do crabs live in the wild, the red king crabs? How long do they live in the wild?
That is a good question, and it's a hard one to answer because we have no way of aging crabs. I've tried to ask some of the female crabs in the lab, and they always get really crabby when I ask them how old they are. That was a joke. The best guess for red king crab is that they can live more than 20 years.
Great. Wow, that's a long time. And then Shivana was wondering how many crabs were used in the experiment that you were showing the video of.
In that one, we started off with 60 crabs per pH treatment, 3 pH treatments, so 180 crabs.
And is that a typical, so one of the questions that was also asked was, how many crabs have you looked at? So is that a typical number of crabs in an experiment? And then how many experiments do you do typically in a year?
So yes, the higher the sample size, the more crabs you have, the better data, the better the data is. That's kind of a universal scientific, I won't call it a law, but principle there. 30 is what we like to aim for the juvenile experiments. As the crabs get bigger, they need more space, and so we often reduce the number. I don't like to drop below about 15 crabs for most of these experiments. For the larval experiments, we'll often stock several thousand for each treatment.
Wow, that's a lot. And then we have a couple of questions about the very small crabs. So Jillian was wondering, how do you pick up the really small?
Very carefully. And actually, one of the cool things about them is the crabs are actually, even at the smallest size, so they're 2 millimeters or 1/16 of an inch, even at that size, if you're very careful when you're picking them up, I can pick up a crab and handle it gently without crushing it, because they've got those hard outer shells, the hard exoskeletons.
And then the last question I want to ask before we move on to the next section is that Sarah and Jeffrey were both asking, where do you get the crabs? Where do you get the very small crabs?
So actually pretty cool, we'll be talking about this later on, but we have some partners, the Alutiiq Pride Shellfish Hatchery, which is over in Seward. And we've done a number of experiments with them where we raise large numbers of juvenile king crabs. And those are for other experiments I'll be talking about later. But for these particular juvenile red king crab, we had some from their work, which we were able to use. Some of the other juvenile crabs we will capture directly in the wild. So the Tanner crab that I showed you, we captured that here in Kodiak using a special net from the wild when it was, again, very, very small.
Wow, that's pretty cool. All right, well, I know that we have a lot of material to cover, so maybe we should go on to the next section and have Switgard come on.
Sounds good.
Okay. So to support our ocean acidification research and measure the changes in ocean chemistry, both in the experiments that Chris just showed you, and outside in the ocean, the Kodiak Lab is home to an instrument called a Berculator. And you see that in the picture there. There are very few Berculators. I think there are less than 10 that are spread out along the West Coast. I don't know the exact number. We have 3 of them in Alaska. One is in Seward, one is in Sitka, and one is in Kodiak here. The Berculator is the most advanced technology currently available for taking the parameters that are involved in ocean acidification. It is not as simple as measuring the pH of the ocean. We also need to take into consideration the alkalinity, which describes how the buffer capacity of the water. You might say how resilient it is to change. This depends on how salty the water is. We call that the salinity, and the temperature, and also the different forms of carbon in the water. So in the top panel there on the graph, you can see how much CO2 is in the surface water. And the graph covers about 8 months of the year. And you see that in the winter time, we have the highest CO2 concentrations in the water. In the middle panel, you see the salt content or salinity of the water. And that usually depends on how much rain you get, and then the rivers swell up and bring more fresh water into the ocean. And that all affects the salinity. In the bottom panel, you see the temperature, and SST stands for sea surface temperature. Our Berculator has a seawater intake line that pumps water from the ocean to the instrument. All of those parameters that are involved in ocean acidification are continuously measured by the Berculator and are used in all of its calculations. Prior to the use of Berculators, science was limited to collecting individual water samples and measuring these parameters one at a time. Now we can see what they are all of the time, night and day, every day, and see how all of these parameters change over the seasons of a year, how different one year looks from another, and what is characteristic for each location where we have a percolator. Now we have a large network, and all the data are or will be shared with open access on the internet. Okay, I think a video is a little bit jerky, but that's okay. We can just advance forward to the next slide. So in addition to these continuous measurements, we can use the percolator to process water samples that are collected somewhere else and sent here for analysis. Kodiak Island is a very unique place in terms of its oceanographic conditions. On the southeast side of Kodiak, we have upwelling of deep water that is very old and rich in CO2. While in the north, we have Shelikof Strait with water that traveled along the coastline and picked up glacial meltwater. Add to this the high phytoplankton productivity typical of Alaska coastal regions, and we get a lot of variation in ocean chemistry between locations around the island. To collect samples from around Kodiak Island, NOAA collaborates with the Kodiak Area Native Association and utilizes a network of tribal environmental samplers in 4 native villages. The samplers regularly collect water samples as well as temperature and salinity and a few other measurements. So we have samplers in the villages of Yusinki and Port Lions and Old Harbor and Larsen Bay, which are those little stars you can see. The green star is where we are, Kodiak. When I get the samples, I process them on the percolator in the order they were received. Twice a year, I report the data back to the environmental samplers and their communities. We also use a percolator to process samples in-house from our own experiments. And another branch of research we're involved in is with the growing kelp industry in Kodiak. Oh, I'm too fast. Kelp, as it grows its fronds, uses a lot of carbon. Just like the plants on land, the kelp uses photosynthesis to absorb CO2, releases the oxygen, and splits off the carbon to turn into sugars for growth. We know this in theory, but nobody has ever measured and documented it in this environment. We don't know how much difference it makes at the site of the kelp farm and in a bigger context. Our research will provide data to discuss questions like, can kelp farming help reduce ocean acidification locally? Are kelp farms good places for shellfish and crab to grow? Perhaps there could be ocean farms in the future where we not only grow seaweed, but also mussels, oysters, crabs, urchins, and other seafood edibles. So from being part of a large monitoring effort all along the Pacific US coast and the Canadian coastline, and comparing samples from around the Kodiak archipelago to learn more about a local oceanography, to forecasting the best growing conditions for crabs, we still have so much to learn about ocean acidification. With the percolator, we can produce the measurements to begin to understand some of these bigger processes that are driving big changes in our oceans.
All right, thank you, Switgard. It looks like we did have a question from Theodore, who is wondering, why are there so few percolators?
Oh, the percolator is relatively new technology, and it is very expensive to get a percolator. Each of the percolators at this point is an individual unique instrument. So each one is a little bit different than all the other ones. So it takes a big grant to get a percolator, install a percolator, and build a program around it to have all the science behind it.
Yeah, that's really, it seems like you can do a lot of things with the percolator. I have one more question, and I know that we have another section to get through, so I'd like to move on after that. But Oliver was wondering, why is there more carbon dioxide in the winter months?
Well, I talked about how the kelp takes up the CO2 and splits it and uses the carbon in its growth, and the oxygen goes into the water at that point. So the reason we have more carbon dioxide in the surface water in the winter is that there are no plants, no phytoplankton using the carbon dioxide.
Oh, that makes total sense. Yeah, that I could see where if you had a lot of plants in the water, that would take up some of the CO2. Great. Well, maybe we should go on to our next section because we have about 15 minutes left, and then we can come back at the very end for more questions.
Okay, sounds great.
So one of my favorite parts of this job that we get to do is to go diving. And the reason we dive is really because our animals live under the water. And so whenever you bring them up into the laboratory, they change their behaviors. They don't act like crabs do in the wild. And so to understand how these animals actually live, how they grow, what they do in the actual habitats where they live, we have to actually go down there and study them. And that requires putting on something that lets me breathe underwater. I've been trying to develop gills for a number of years and just not gotten the slits on my neck yet. So until that happens, we're going to still continue breathing compressed gas underwater. So one of the coolest projects that we've been involved in here has been the ACCRAB program, which is the Alaska King Crab Research, Rehabilitation, and Biology Program. And this was started long before even I got to the lab. Basically what happened is there used to be a ton of, a huge population of red king crab in the Gulf of Alaska. But in the 1970s, that population crashed out. The fishery has been closed for, I think, going on 30 years now, and we have not seen any recovery of that. So one of the ways that people are interested in trying to help red king crab recover is by growing up juveniles in a laboratory or a hatchery. A single female red king crab can have, on average, about 250,000 babies every single year. That's a lot of babies. And in the wild, they're very small, and only about 0.1 of them, probably less than that, actually survive to become juvenile red king crab. But in the hatchery, we can get that up to somewhere maybe about 30%, which is a lot better. So what our laboratory has done is, in conjunction with university partners, as well as with the Eluctic Fried Shellfish Hatchery and some of our industry partners, has been to develop a program where we look at developing the technologies to rear up the crab. And that's what the Eluctic Fried Shellfish Hatchery does. And then our side is to look at what happens to those crab after you release them. Because obviously we don't want to just put out crab out there if they're going to become expensive fish food. So what we do is we take those crab out and we release them into the natural habitat. So the picture on the left down there is actually a jar full of baby crabs, which we are about to scatter onto the substrate down there. And you can see that there's tons of shells and rocks and cool stuff like that for them to hide under, because at this stage, that's their best defense is a good defense, is they hide under those shells, and then hopefully the things that want to eat them can't find them. And then we have to go back out and we have to find those crabs again. So on the right-hand side, you can see a diver who's got actually found one of the little baby king crabs, is holding it right there. And in the next slide here, we'll actually see a video of what this looks like. So in order to count them, we bring this plastic PVC frame under, and that way we know exactly how much area we're searching. And then you go through that with a pair of very long pair of tweezers there, and you pick up and you have to flip over every single rock and every single stone to see if, like on this particular shell, if there might be a crab underneath it. And that way we can count the number of crabs and we can figure out how well they're surviving. We can compare it to other populations of king crab, where we have estimates for healthy populations, and see whether or not this is working well enough that it's worth trying on a kind of on a bigger scale here. So on these types of experiments, we're typically releasing somewhere in the hundreds to about 10,000 crabs. Obviously, if you're going to be doing this on a recovery scale, you would have to bump that up to millions of crabs. So this is a way of doing kind of a small scale experiment to determine whether or not this is a feasible thing to do underneath there. So you can see whoever the diver was on that dive recording the king crab that they found, and then they'll be moving on to the next plot in a second here. I'm not sure how much more video we've got on here. So, oh, I think we've looped back around. There we go. So will we do questions on that at the end?
I think so one of the things that Allie had wanted to get our audience's help on is to name our next octopus at the Kodiak Lab, because they used to have an octopus at the lab, but they had to let it go because it got too big. So what we're going to do is we're going to launch a poll, and if you guys would like to put your vote in for the next Kodiak Lab octopus, you can just select one on your screen there, and then we can go ahead and close the poll after about 30 seconds. So Chris, do you want to tell us a little bit, or Allie, do you guys want to tell us a little bit about the octopuses in your lab? Because I think that Katya was asking whether you still have an octopus at the lab, or whether the octopus had to be let go.
So the one we used to have in the lab did get let go a couple months ago because she just got too big, or he got too big. They will outgrow the tank. These guys can get upwards of 20 kg, which is more than 44 pounds. This is one of the things I meant to mention during the diving thing, is we do science underneath there, but we also take the opportunity to collect animals while we're under the water for the touch tank, for the Ocean Science Discovery Lab, and most of the octopuses we've found over the years have been captured by our divers here. So we've in our laboratory, we have done actual research studies on octopuses. They're one of the federally managed species, both in the Gulf of Alaska and in Bering Sea. And so one of our other researchers here, Christina Conrath, did a big study a few years ago looking at octopus growth. And one of the cool things about that project was that, so octopuses are notorious for being masters of escape. And so in order to keep them from escaping, and literally what the octopus would do would be to crawl out of their tank, crawl into the crab tank next door and eat the crab, and then go back home. If we didn't do this, is actually get plywood, put it on top of the tanks, and then clamp it down on all four corners, in order to keep those octopi in there. So
that's really great. So I just wanted to share the results of the poll. It looks like we had a close race between the first three options, Ophelia, Squirt, and Cece the cephalopod. Hippolyta was a little bit behind there, but it looks like Squirt won for the results for the name of our next Kodiak Lab octopus.
That is an awesome one, because one of the fun things is when we bring kids back to actually, interact with the octopus, they can, get in and shake a tentacle or two. But very frequently, and this has happened to my own children, is they will squirt right out their excellent siphon, which they use for jet propulsion in water, and get everybody wet. So a big octopus can splash a very large squirt, a large quantity of water. So
that's great. Well, we have about 5 minutes left, so I was wondering, Allie, whether you could hide your slides and whether you guys could answer some questions.
Yeah,
So,
let's see here. I'll turn those off. Thanks
Great.
so much, Lisa. They're going to push the camera back. Svigard and Chris are actually in the library together, but they're going to maintain their distance and wear their masks. But yeah, we are available to answer any questions that anybody has.
So Kip was wondering, don't octopuses only live for about a year?
Giant Pacific octopuses live till, I think, about 4. Chris can correct me, but I believe it's about 4 or 5 at max.
Yep.
And then Shante was wondering, Sean from DC was wondering, how do half of the king crabs die after they're born?
So there's a lot of things that likes to eat king crabs. So when they're born, they're actually tiny little shrimp-like larvae, and they start off maybe a millimeter long, really, really small guys. And they're floating in the water column. And while they're in there, everybody likes to eat them. Everybody likes to eat crabs. And so they get eaten by salmon, they get eaten by pollock, they get eaten by all sorts of things that are floating in the water column, or fish that are in the water column eating these guys. And a second thing for king crab that's really important. So the habitat we put them in is that nice shelly bottom, right? But there's relatively little of that habitat in Alaska. Most of the bottom in Alaska is soft, sandy, or muddy, and it's flat. And there's nothing for crabs to hide under. And so for a juvenile crab, it takes about 3 to 4 months in the plankton where it's floating around and kind of swimming. And then it has to find the perfect habitat. And if that crab, if the larvae doesn't find that perfect habitat, almost as soon as it lands down there and becomes a crab, something's going to eat it. So because there's relatively little of that habitat, a lot of those crab larvae aren't going to find the right habitat, and they're going to get eaten by something that way too.
Right, that makes sense.
Yeah, Lisa, too, I was just going to mention, if people have specific questions about crab or they're more interested about crab, they can go back and watch our NOAA Live Alaska that Leah Zacher and Aaron Fidawa did in November or October, I believe. And they talked specifically about crab, and it was a really cool webinar that they should check out.
That's right. And we have a link to that webinar on our NOAA Live Alaska website. So Theodore actually had a question about crabs and was wondering, how do the people not get pinched by crabs in the petting tank?
Yeah, it happens, unfortunately. We just try to warn everybody when they come in. You got to watch out for your fingers. So when you're in the touch tank, it is an enter at your own risk type situation.
And then Katya was wondering, do you guys tag fish or crab and then let them go?
Yeah, we do. Leah Zacher actually has a project that's going on right now, and she tags crab, big red king crab in the Bering Sea, and then she tracks them with sail drones. It is a really, really cool research project, and I think there might actually be a series of blog posts on the NOAA page that maybe Lisa could link to as well. It's a very cool project.
Yes, and actually I did put a link to Leah's science blog. So a blog is a science blog from us is something where the scientists, when they go out to the field, they write something about what they're doing each day and put it up as a post on our website. And so I linked to that on our NOAA Live website as well. I do have a last question for Switgard. And Kaylee was wondering, how do you get a borculator and how much how much does it cost?
Well, it's not personally my percolator, but you basically, when you do big research like that, you have to write a proposal and you have to find the money for the research. And then you write basically what you want to do with it, and you have to ask for that money. How much does it cost? It costs about as much as a small airplane.
Oh, wow, that's pretty expensive. So one last question for the three of you is, if you had one piece of advice to give our students and our viewers, if they were interested in doing the kind of research that you are doing, what would it be? And maybe we could start with Allie.
Yeah, that's a really good question. So my piece of advice would be to get involved early, get involved through your school, ask your teachers for help, ask them for support, and you will be rewarded. So I actually started as a high school student here in Kodiak with the high school program and volunteered at the same lab. So taking the initiative when you're young, learning from your mentors, it's really key.
Great, thanks, Allie. Switgard, what would you say?
I would say don't get discouraged. You will have to make a lot of tries, and a lot of times you're running into walls, but you always have to find a way to follow your heart, follow what you want to do, and eventually life will give it to you.
Great, thank you, Switgard. And Chris, you got what advice would you have?
For students who are interested, I would say study hard. It's math and science are of course really important, but in addition to that, Switgard talked about writing grants, and a very large proportion of what I do is writing papers and writing grants. And so learning to write and communicate well is super important. So don't math and science, absolutely. Don't disregard the English, your English class either. So.
Great. Well, thank you guys so much for giving us such a great tour of the Kodiak Laboratory, and thank you to our audience members for joining us on this tour. And we have one more tour in the NOAA Open House Series tomorrow, and I hope you guys can join us. But thanks very much for coming all the way to Kodiak with us today. Thanks very much. Bye-bye.
Thank you so much, everybody.
Thank you.
Thank you.